Welcome everyone again to the 4th annual MCHRI Symposium. And welcome to session number 1, Emerging Maternal Perinatal Health Discoveries. We have 3 speakers this morning, and we'll go ahead and get started with the first speaker. Please feel free to add your questions to the Q&A, and I will help moderate questions at the end of each talk. So our first speaker is Dr. Nima Aghaeepour. He is an assistant professor in the Department of Anesthesiology, Perioperative, and Pain Medicine. He is an active investigator in the March of Dimes Prematurity Research Center and is doing phenomenal work bringing his data science expertise to the realm of perinatal health. So with that, I will turn it over to Dr. Aghaeepour.
Thank you for the kind introduction, and thanks for having me here. I'm going to speak about preterm birth primarily. As Dean Miner mentioned, prematurity is currently the single largest cause of death in children under 5 years of age, both in LMICs and in high-income countries. I'm going to speak about our immunological studies looking into preterm birth, but we'll also speak about social determinants of health and how they interact with biology, and finally about our most recent work in using AI to redefine the taxonomy of prematurity. I'm representing a large group, including a large number of trainees and staff in our group, as well as David Stevenson, Carl Sylvester, Gary Shaw, others who work through the Prematurity Research Center and MCHRI, our partners at various biorepositories in Southeast Asia and Africa, the Bill and Melinda Gates Foundation that has been more of a partner than just a funding agency, as well as other funding agencies, Martin Angst and Brice Goudelier, my co-PIs, especially in the immunological studies. I'm going to go over a relatively large number of projects that we're quite excited about, and you can take a screenshot of this page and go to the poster session and look at the 9 or so posters that describe the work that I'm going to give you an overview of in details, and I will put this slide back up at the end of the talk. As this audience very much understands, and as Dr. Molly just mentioned, what happens in those first few months of life have significant consequences for not just child health, but also for sequelae that extend well into adulthood. But my lab is focused on understanding developing a holistic understanding of pregnancy and early life from a variety of views. And in doing that, we use biological modalities and as well as other tabular data like lab tests, medications, but we also use more complex unstructured data sets, including imaging data, live stream hemodynamics data from the intensive care units, and so on. And in the long term, we are planning to integrate all of these various sources to develop a holistic understanding of maternal and child health, rather than trying to just come up with diagnostic tests for one condition at a time, because all of these are in the grander scheme of things related to each other. I'm going to talk about some of these integrations over the course of this presentation. First, let's dive into our immunological studies. During the course of pregnancy, the immune system strikes a balance between pathogen defense for the mother and immune tolerance for the fetus. And if we can understand the normal dynamics of pregnancy of the immune system during pregnancy, we can use that to detect abnormal changes during pregnancy. In our first study, we looked at a cohort of normal pregnancies at Stanford. These were not preterm, and we used the state-of-the-art mass cytometry platform to measure hundreds of thousands of individual cells from the immune system for every patient at every trimester of pregnancy. This allowed us to measure a large number of cell types and, more importantly, functional markers inside each of those hundreds of thousands of cells. These went through fairly standard machine learning. You have your gestational age and your data, you're minimizing your error rate. And of course, we were able to leverage work done by leading Stanford statisticians who help make these models easier to interpret, easier to validate, and help them to better account for the highly correlational and highly modular nature of the immune system. That allowed us to develop a fairly robust model of immune changes during pregnancy, and we were able to validate that in an independent cohort. This was just the immune system's characterization at a single cell level. But we were also able to, through the Prematurity Research Center, we were able to leverage the expertise of various leading laboratories at Stanford. For example, the work that Dr. Relman is doing with the Gates Foundation to characterize the microbiome, the work from the Quake lab on cell-free transcriptomics, and all kinds of other technologies that are available on campus to us. We were able to characterize those same cohorts of women using all of these various technologies. And when you have a unique data set like that, the first question that you want to answer is, well, what correlates with what? And it turns out that when you subject biological systems to something as significant as pregnancy, a lot of biological measurements are going to correlate with each other. But we have to take a more systematic machine learning approach here. But taking all of this data and just putting it into a standard off-the-shelf machine learning algorithm doesn't quite work, because these data sets are very different. Some of them are measuring many various biological measurements, and others are carefully designed small but targeted assays. And accounting for size alone is not enough. Here is a large data set. Even though it's large, it's fairly simple. You only see 3 clusters. And here is a smaller data set. And even though it's much smaller than the other one, it's a lot more complex. So much of the research in my lab is focused on developing the right approaches to take each of these modalities, extract the information that's needed from them, and then have a higher level model, put everything together to make the final predictions. This gets even more complicated in multi-site studies. In one of our early works with the Gates Foundation, we looked at a case control study of preterm birth from 5 different LMIC sites. And we quickly learned that we need to build models not just for each modality independently, but also we need to build models for each of these sites and then take that model and optimize it using data from the other sites. This is to honor the environmental and genetic differences that we see all over the world, but also to acknowledge that there are shared biological pathways between these different sites that can be leveraged to build universal models. Once we did that, we were able to predict preterm birth using biological samples collected at the first trimester of pregnancy fairly accurately, subject to appropriate cross-validation, of course. And the model was picking up the placental growth factor in plasma, right to the right of it, GCSF, PD-L1, IL-6, IL-1RA, a classic textbook module of inflammatory proteins in blood. And right next to it, we have a module of metabolites in urine. And this points to one of the advantages of data-driven studies like this, in that we can build low-cost surrogates for our complex models using just urine samples that allow us to capture the more complex plasma and immune system measurements so that we can better scale them up to LMICs. This was a really large and collaborative effort, that's why I'm showing you the author list, to acknowledge our partnership with various biorepositories all over the world, as well as the Gates Foundation. And the next step for us, which we have already started working on, is to scale this up to 10,000 pregnancies from these biorepositories, and to simultaneously integrate biological factors with epidemiologic factors to understand how these models can be made more generalizable, and to identify non-pharmaceutical intervention targets as well. We have repeated the same strategy now for preeclampsia, as well as a number of other complications in pregnancy. And you can take a screenshot of this and look at either the poster session or the various publications to learn more about them. The next part of the talk is about social determinants and biology. In our prematurity research center cohort at Stanford, we administered stress questionnaires to these women at the first trimester of pregnancy. And in a recent publication, we showed that we are able to predict various pregnancy-associated pathologies using the stress questionnaires alone. These are just a subset of the complications that we have looked at. And in our follow-up work, we showed that this stress data is actually connected to the multiomics data that we have been able to measure. The direction, the causality of it is not known to us yet, but there certainly are models that can include both biology and stress measurements to predict various adverse pregnancy outcomes. And this points to a very exciting opportunity to potentially use modifiable factors, including various aspects of stress, to steer biology in the right direction. Similarly, using wearable devices outside the context of maternal and child health, we have been able to link physical activity and sleep to changes in the immune system. And just very recently, we repeated the same study in a cohort of 1,200 women in collaboration with WashU, where we had a smartwatch measure their physical activity over the course of pregnancy. And we built a quote unquote clock of physical activity and sleep that changes with gestational age. That part is not too exciting, but the more exciting part is that the outlier patients, those who are not following the general trends of physical activity and sleep over the course of pregnancy, end up being significantly enriched for morbidities, including preterm birth and preeclampsia. And again, this points to an exciting opportunity to use interventions that target physical activity and sleep to steer biology in the right direction to prevent adverse outcomes. I'm sure many of you have seen the body of literature on the connection between sleep, physical activity, and inflammation. And I just told you about inflammation and preterm birth, but we are quite excited about the potential to put all of these together with our colleagues at WashU to design an interventional study soon. All of this work is enabled by a wide range of machine learning algorithms that we develop. Most of these tasks cannot really be achieved using standard machine learning algorithms. I don't have the time to walk you through all of them, but it's not just algorithms that we develop ourselves. We also work with the entire machine learning community through crowdsourced machine learning challenges, where we make data sets available to a wide range of machine learning scientists who otherwise wouldn't get exposed to maternal and child health. And that helps with training the next generation of scientists. I will show you one example of the type of machine learning work that we do. I'm sure you all are familiar with the Pearson correlation, which was created about 100 years ago. And since then, we have come up with electronic health records and multiomics studies that are measuring hundreds of thousands of variables. And we still build correlation networks using the same equations that were created 100 years ago. So some very smart people in our group, Huda and Martin, came up with a new formulation of the same correlation concept that allows us to measure correlation networks much faster than what was possible before. So large data sets that wouldn't even fit on high performance clusters can now be simply constructed on a laptop, and calculations that used to take days and weeks and months are again now taking only hours, not even days, on a simple laptop. We are quite excited about this because for the first time, it allows us to build really large networks on our multiomics data sets. And instead of just asking what proteins or genes go up and down subject to a particular disease, we can now ask about changing dynamics, changing of correlations, the two previously correlated biological modules that are no longer correlated if a particular disease is happening. And this is helping us unlock a completely different layer of biology in these data sets. And finally, the last example that I will show you, because I was told I have a couple of extra minutes, is our work on redefining prematurity itself. Everything that I showed you so far is about the connection between multiomics data and preterm birth, which is gestational age at 37 weeks. But as a computer scientist, I couldn't even remember the number 37 or understand why gestational age. I assume there is a medical reason for that, but it turns out that there really isn't. It comes from mortality rates that come from historic population level studies in Europe several decades ago. And mortality rates themselves come from neonatal morbidities. And this, I'm sure, already looks like a neural network to many of you. So we simply took the unique OSHPAT and CMQCC resources that we have in the state of California, and we used a neural network to connect clinical data to neonatal morbidities directly, circumventing this traditional definition that's based on gestational age. And we ended up doing significantly better than gestational age. This is a model built on 1 million pregnancies, tested in 10 million pregnancies. This is gestational age, that's preterm birth. This is extreme preterm birth. This is a small for gestational age. So we are doing way better than traditional taxonomies, but our precision and recall is not anywhere near the level that I would expect before I talk about precision medicine approaches. So the next part of this study was that we introduced biology to the system. We had metabolites collected from dry blood spots on the neonates themselves, and we plugged that into our model this time, and our precision recall became a lot better. And through some computational tricks, we were able to push it even higher. So the consequences of these are quite significant, that for something as challenging as necrotizing enterocolitis in red, using dried blood spot samples taken at birth, we are able to predict this. And this can potentially change the way that we treat necrotizing enterocolitis in these babies, especially for the babies that are born too early. And the last piece of the story is that we were also able to integrate all of these with Stanford's electronic health record, because uniquely we have long-term maternal history on many of our patients, and we have the newborn electronic health record within the same institution. This allowed us to deploy really advanced AI techniques to try to predict various neonatal morbidities. As Dr. Molly was telling us, maternal health is directly related to child health. And again, we have really exciting results. Unfortunately, I can't tell you how these work yet, but feel free to send me an email and we'll have a one-on-one conversation about what goes into these models and how they work. But the results are quite exciting. Again, for something like necrotizing enterocolitis, we get remarkable area under the curve, and our precision recall is 40 times higher than random. More importantly, these models start working often several months before birth, before the baby is even born, and they continue to monitor the electronic health record and other data modalities after birth as well, and they continue to become more accurate. And again, I'm not going to go into the details of the model, but it's looking at a wide range of variables that are being collected at Stanford. And the last piece is that as I already told you, the precision and recall of clinical factors and electronic health records at the end of the day is not enough to enable precision health. So what we have most recently done is that we have integrated our AI-based model of the electronic health record with data from the Metabolic Health Center at Stanford, because again, uniquely, all of these are under the same umbrella at Stanford and connected to each other. And through that, we were able to show that for many of these neonatal morbidities, the connection between our model and objective biological data from the Metabolic Health Center is stronger than the connection between biology and individual ICD-10 codes that you can get from these data sets. And that is quite exciting, and a direction that we are headed to in the future. And of course, the biological pathways that are coming out of these make a lot of sense too. In the long run, our aim is to use cutting edge decision intelligence systems to build a platform where neonatologists and OBGYNs can interactively work with this AI model and fill the knowledge gaps that would allow the model to make even more accurate predictions and guide therapeutic decisions. In summary, I told you about our immunological studies, about modifiable factors, machine learning, and our new taxonomy for preterm birth. As promised, here are the posters that you can go look at again. And thank you very much for your time.
Oh, Nima, thank you so much. That was an amazing tour de force. And it's so exciting to see the direction it is all going. I have to say one thing that struck me was understanding that when we enter things into the electronic health medical record, the importance of entering it as correct and accurately as possible, so that the use of the EHR can be really used in this incredibly powerful way. Nima, if you want to go back one slide so your posters are there, we can leave that up while I ask the questions. We have a number of questions coming in. What do you consider are the roadblocks for connections to integrate EHR and AI modeling? I guess besides accuracy of data entry.
Oh, I'm not too worried about accuracy of data entry, because as I showed in this slide, the connection between our AI system and biology is stronger than individual ICD-10 codes on the y-axis. So even if some of these codes are incorrect, the models are these days looking at tens of thousands of variables, and they can figure out that, all right, something here doesn't quite look right, and they can ignore that. But the more important thing I would say is building the scale that is needed to build large-scale biological data sets, because these are currently very expensive, and at a place at Stanford, the Metabolic Health Center can do some of that, but then to scale that up to other institutions and other settings, it will take a lot of resources and innovations. As I showed, our clinical only or EHR only models, as impressive as they are from a statistical point of view in that everything is significant, they are not at the level that is needed for them to enable precision health. We do need to introduce biology into these in the cases that are non-obvious, at least in the marginal cases. We can use our AI platforms to make the trivial decisions that are easy, the cases that are not too difficult, but for the ones that are kind of on a spectrum, we do need to introduce biology into them, and the scaling up these expensive assays, I think, is one of the major barriers to entry.
Great, thank you. And can you talk more about the social determinants and how you calculate the variance? And what are the next steps to prioritize those with the biggest effects?
Yeah, so that goes beyond my area of expertise. What I can tell you is that when we look at stress, there are specific aspects of stress that are connected to biology and through biology to various maternal outcomes. But then we are working with various experts, including from the psychiatric department, to figure out what are the right interventions for those. Similarly, with physical activity and with sleep, we are working with the appropriate experts at Stanford and WashU to design the right way to intervene in a safe and responsible way. And the electronic health record model that I showed you at the very end, I didn't go into the details of it, but that model is also, without our intervention, picking up several socioeconomic factors that are key players in the families that are key players into neonatal morbidities. And again, I think there are significant opportunities for interventions there as well.
Great, thank you. And then based on the keynote address that we heard, what are ways your research can connect to sexual reproductive health?
In many ways, that's quite interesting. We are, from our Stanford work alone, we are seeing various opportunities for intervention so effective medications that are used for sexual reproductive health on pregnancies are well documented, but we are seeing connections between some of these medications, including some that were mentioned on the keynote. We are seeing the connections between their use in the mother and the specific neonatal morbidities. I think there are opportunities there to better study the impact of these medications, whether protective or harmful on the babies. And similarly, in our work in LMICs with the Gates Foundation, again, we are seeing direct connections between sexual reproductive health and preterm birth, and that has been documented before in other settings as well. I think there are opportunities to do a better job there.
Great, thank you. And I think we'll take another question. Where do you see connections with industry benefiting your research?
There are two aspects to this. One is better data availability, and second is better data analysis. In terms of data availability with electronic health records and population level data sets and so on, there is a lot of work that is being done at an industrial level. And more importantly, at a biology level, proteomics assays that cost $1,000 per sample today, there are several startups in Silicon Valley alone that are planning to bring that cost dramatically down in the near future. I think ultimately the industry is going to be successful at somewhat in bringing that cost down, but we are not going to, in the short term, get to a place that these biological assays can be used broadly for everyone. I think we will need to still deploy AI systems that do a first level of monitoring and then pass on the prediction, pass on the patients that require further biological characterization to new technologies that are being developed at Stanford and in the industry.